Good afternoon. I'm Sofia Kyriazopoulou, and I will present Kiled, a new method for relationship inference and its applications to the detection of previously unreported relationships in 4 HapMap populations. So I will motivate the question we are trying to answer with this toy example, which I will be using throughout the talk. Assume that we have genotype John and Dave in a set of genomic locations, and we want to use this information to infer the relationship between them. For example, we want to decide whether John and Dave are first cousins or whether John is the great uncle of Dave. There are several settings where similar questions arise. Forensics is an obvious application, but other interesting applications include personalized medicine or association and linkage studies where unspecified relationships can seriously affect the statistical results of the study. To follow the rest of the discussion, it's very important to understand the notion of identity by descent, or IBD. 2 genomic regions are called identical by descent if they're inherited from the same common ancestor of the individuals we are considering. In this toy example, and assuming that these pairs of blocks here are pairs of homologous chromosomes, you can see that John and Dave have these regions between the vertical bars IBD. Different types of relationships imply different underlying processes of meiosis and recombination, which in turn lead to different patterns of IBD along the genome. So how can we model this IBD process? And how can we use this information to answer the question at hand, which is to infer the relationship between 2 individuals? Previous methods, such as RelPair and PRES+, have used hidden Markov models, or HMMs, to model the IBD process. An HMM for this application would basically transition between regions of the genome where the 2 individuals share 0, 1, or both copies of their genome IBD. The transition probabilities depend on the exact relationship that we are modeling, while the output of the HMM would then be the likelihood of the observed genotypes under the particular relationship, together with a set of posterior probabilities of IBD along the genome. However, these HMMs do not capture the whole picture. For example, consider the first pair of pedigrees here at the top. These are obviously not the same pedigree, but from the point of view of the HMMs of the previous slide, they're exactly identical. We could swap the genotypes of the 2 individuals, and the likelihoods computed by the HMMs will be exactly the same. Similarly, the 2 HMMs here, the 2 pedigrees here at the bottom, can hardly be distinguished by those HMMs. And the reason is that the transition probabilities of the HMMs depend on the number of branches between the 2 individuals, not on the exact pedigree configuration. However, as you can imagine, there are cases where we do want to distinguish between those pedigrees because they're obviously not the same. We call such pairs of pedigrees rotations because you can think of them as resulting from rotating the pedigree around the axis of the common ancestors. So our goal with Kiled was first to create a model that can consider a much larger set of relationships than previous methods that only considered a handful of different alternatives. And secondly, and most importantly, to allow our model to distinguish between relationships where previous methods had very poor performance. And this is exactly the rotated relationships. To achieve these goals, we started with HMMs that are similar in concept to those used by previous methods, but instead of just relying on the computed likelihoods, we also considered additional features which led to an improved accuracy. For example, the genome-wide IBD sharing between 2 individuals differs between relationships of different degrees. For example, full siblings are expected to share about 50% of their genome IBD, while first cousins on expectation will share about 12% of the genome IBD. The number of transitions between IBD and non-IBD regions differs even within the same degree. And the reason is that, as I said, the recombination pattern differs between relationships. Finally, the haplocounts capture linkage information which is completely missed during the computation of the HMMs and help us distinguish between rotations. I will define the haplocounts in a couple of slides, but before doing that, let me explain how we generalize the HMMs of previous methods in order to consider a much broader set of relationships. The key observation here is that all relationships between non-inbred individuals must fall into exactly one of the following three categories. Relationships with two common ancestors, such as full siblings or first cousins, relationships with one common ancestor, such as half-siblings, or relationships where one of the individuals is the ancestor of the other. The only thing that differs between the relationships within each of these categories is the exact number of generations between the common ancestors and the individuals we are considering. Therefore, we can create a template HMM, one for each of these categories, and then parameterize it with a number of generations between the common ancestors and the individuals we are considering. I will now define the haplocounts as I promised, which help us distinguish between rotations. As I said, the HMMs I just described cannot distinguish between rotated relationships, and the reason is that they assume that the underlying genetic markers are unlinked. However, this is not always true, and there is actually information in the linkage pattern that we can use to distinguish between relationships. Let's consider the same toy example I showed before and assume that these pairs of blocks here are a pair of homologous haplotypes of the two individuals, of John and Dave, and the bolder segments are segments inherited from a common ancestor. Of course, in practice, we cannot really observe that, but what we can infer is that the region between the vertical bars here is IBD between the two individuals, while the surrounding area is not. Now let's look at a small window around the transition between IBD and non-IBD. As you can see, in this region, John has a consecutive haplotype coming from the common ancestor, while Dave has a mixed haplotype coming from more than one ancestors. Therefore, you could say that Dave has a break in the linkage pattern of the common ancestor. And since the further away we move from the common ancestor, the more likely it is that such a break occurs because there's more chance for a recombination. Dave is more likely to be more distant from the common ancestors than John, as in the case of the second tree. To formalize this intuition, we define the haplocounts. A haplocount of an individual, say John, in a region around an IBD transition is basically just a count of how many times in the reference population we see this haplotype. To account for genotyping errors, instead of just adding 1 for each haplotype in the reference that matches the haplotype of John and 0 for every haplotype that doesn't, we instead consider the probability that a haplotype in the reference is transformed into the haplotype of John through genotyping errors. So in this formula here, H of I is the ith reference haplotype, H of John is the haplotype of John in the same region, and then F of epsilon is the probability that the two haplotypes look the same due to genotyping errors. We then sum over all the haplotypes in the reference and divide with the number of such haplotypes. We compute this haplocount for both John and Dave and for all the regions in the genome where there's a transition between IBD and non-IBD. And then we define this ratio R, which is just the fraction of such transitions where the haplocount of John is greater than the haplocount of Dave. So the intuition here is that if R is much larger than 0.5, then John tends to have much larger haplocounts than Dave. Since the haplocount of John is a measure of similarity between John and the reference population, in that case, John is more likely to be closer to the common ancestors than Dave. The other way around, if R is much less than 0.5. If R is around 0.5, then we could say that there is no evidence that one of them is closer to the common ancestors than the other. Having defined the haplocounts, I can now move back to this diagram I showed before and describe the full Kiled procedure. As I said, we start with HMMs for several relationships from which we obtain the likelihoods of these relationships. From the HMMs, we also obtain a set of posterior probabilities of IBD along the genome, from which we can compute these additional features. Then all these features are input into a multi-class linear classifier, whose output is then the predicted relationship between the two individuals. To evaluate CAROT, we started with simulated data where the underlying truth was known. We obtained genotypes from 2 HapMap populations, and after eliminated individuals that were known to be related to each other, we divided the remaining individuals into 2 completely separate groups. One group that was used as our reference population to compute allele and haplotype frequencies, and another group that was used to simulate pedigrees for different types of relationships. And then the simulated pedigrees were divided into a training group that was used to train our model and a test group that was used for evaluation. I will start with a comparison between CAROT and 2 other methods, RelPair and PRESS+. These are both maximum likelihood methods. That is, what they do is that they run HMMs for different relationships and then just pick the model that maximizes the likelihood without considering any additional features. Since these 2 methods only consider a very small set of relationships, we restricted this comparison to this set of relationships. The results are shown in this plot where on the x-axis is the classification error, that is the number of pairs of relatives that were classified into the incorrect relationships by each of these models. And the error is shown for each of the relationships considered. As you can see, with the exception of one relationship, CAROT achieved a smaller classification error than the other 2 methods. And here at the bottom is the average classification error, averaged over all the relationships that we considered. And again, you can see that CAROT has a much better performance on average than the other 2 methods. However, this is not the ideal evaluation setting for CAROT, because CAROT was designed to distinguish between rotations and relationships of the same degree, and this set here doesn't really contain any rotations. Therefore, to fully evaluate CAROT, we simulated pedigrees for all possible relationships of degrees up to 5, considering all possible rotations. The results are summarized here, where here the classification error is on the y-axis, and the error is averaged for all relationships of the same degree, and the degrees on the x-axis. For a comparison, I'm also showing you the performance of a maximum likelihood approach, which is similar to what previous methods have been doing. That is, we run our generalized HMMs for all relationships considered and then just picked the model with a maximum likelihood. I'm also showing you a baseline performance, the blue bars, which is just the performance of a classifier which randomly chooses a relationship of the corresponding degree. As you can see, CAROT, which is the red bars, consistently outperformed the maximum likelihood approach, which suggests that the additional features that we considered, that is the number of transitions between IBD and non-IBD, the estimate of IBD sharing, and the haplocounts, do actually play a significant role in distinguishing between relationships. Finally, we wanted to evaluate CAROT on a real dataset, and we used HapMap, which is a widely used dataset, and which by design contains many first-degree relationships. In addition, there was a recent study that used RelPair to identify additional first-degree and some second-degree relationships in HapMap. Visual inspection of these relationships suggested that some of them could be combined into much larger pedigrees, which in turn suggested that there were actually more relationships in this dataset that were missed by the previous study. We therefore applied CAROT on 4 HapMap populations in order to detect relationships of degree up to 4, and the results are summarized here. CAROT correctly identified almost all of the previously detected relationships, and in addition, it identified 68 relationships that weren't reported before, and were mostly relationships of third and fourth degree. Since there isn't really any other tool that can reliably distinguish between rotated relationships and make accurate predictions for degrees higher than 2, it was hard to compare these 68 new relationships with some other method. However, as I said, there were cases where relationships of smaller degree could be combined into larger pedigrees. And to see what I mean, consider the second tree here. Here, all the individuals that have an ID are HapMap individuals that were genotyped, and the individuals that only have a number are missing ungenotyped individuals. So in this tree here, all the first degree relationships, the parent-child and full sibling relationships, were detected before. However, the relationship between the first cousins relationship was not detected before. However, if we believe that the first degree relationships are real, then we should believe that the first cousins relationships is also a valid, true relationship. In this, although it was not reported before. In this way, we were able to verify 25% of our 68 new predictions, and also verify that about 1/3 of them were of the correct degree. Finally, I'd like to show you two of the pedigrees that Caro detected. One of them is the pedigree I just showed you, and another one is a much larger pedigree from the Mexican population. And it is interesting to note that, as I said before, all the first degree relationships in this pedigree were reported before, but the entire pedigree structure was not. And more interestingly, Caro identified the third and fourth degree relationships between the individuals here at the bottom without really taking into account the genotypes of the intermediate individuals, which means that Caro would have identified this relationship even if the individuals in the middle were not genotyped, while previous methods would have completely missed these more distant relationships, because usually they don't even consider so distant relationships. I would like to close by saying that although relationship inference was studied a lot in the past, the advent of genotyping and sequencing opens up opportunities for new exciting applications of relationship inference. For example, in the past, forensics was considered the major application of relationship inference. However, as the HapMap case study suggests, relationship inference can be very useful for detecting unspecified relationships in large cohorts, and therefore it can be useful for association and linkage studies where unspecified relationships can have a serious effect. Additionally, relationship inference can be useful in personalized medicine, for example, because the IBD patterns between related individuals affected by the same disease can give us information about the disease mechanism. I hope I convinced you that Caro is more accurate in predicting the exact relationship between individuals than any of the existing methods, and therefore it can be a very valuable resource for such applications. Caro is available on our website, and I'd like to close by thanking all the members of the Bajogu Lab. Some of them are here in this room, and especially the people who actively contributed to this work, and of course my sources of funding. Thank you very much.